Models of DNA replication. DNA is a double helical structure which transfers genetic information from one generation to next. So, after the discovery of the double helical structure by Watson and Crick, there is a question among the scientists by which model this DNA is getting replicated. So, there is a three proposed model in which the DNA is replicated. That model is proposed by the scientist group. The three models are conservative method, semi-conservative and dispersive method. And here these three methods are given. And first one is the conservative method. In conservative method, among the two daughter DNA molecules, one of the DNA is completely original. That means both the strands, both the double stranded molecules are completely original, whereas in another DNA molecule, both the strands are newly synthesized. This model is called as conservative model. You can see here, both the two strands are here, in which one of the daughter is completely similar to parent DNA molecule. That is, both the strands are derived from parent, whereas the another daughter DNA molecule is completely newly synthesized molecule. So this is conservative mode of DNA replication. And the second model is the semi-conservative model of DNA replication. In this model, you can see, so the two strands of DNA molecules are first it's getting separated, that is unwinding happens. And both the strands are acts as a template for DNA replication. And the newly synthesized complementary strand will be synthesized based on the base pairs which are present in the parent strand. As a result, the daughter strands, in the daughter molecules, one of the strand will be from parent molecule, whereas one of the strand will be newly synthesized one. So this is a semi-conservative. Semi means half, half conservative. So half of the DNA molecule will be received from the parent, whereas the half of the molecule will be newly synthesized one. This is the semi-conservative. And third one is the dispersive model of DNA replication. This is entirely different from first two types, that is conservative as well as the semi-conservative mode of DNA replication. You can see here, two strands are here. From these two strands, two daughter DNA molecules are synthesized, in which both the newly synthesized as well as the daughter DNA molecules are patched up together, are mixed together, and complementary bases are arranged and the two DNA molecules are synthesized. So these are the three proposed modes of DNA replication which is suggested by the scientist group. And now there is a big question. So in which model, among these three methods, in which model this DNA is getting replicated? That is the big question among the scientist group. And that is solved by two scientists, namely Meselson and Stahl. So these two scientists, Meselson and Stahl, after the discovery of double helical structure of the DNA by Watson and Crick, after the publication of their work in the year 1954, these two scientists, namely Meselson and Stahl, these two scientists teamed up together and they worked on DNA replication to find out the mode of DNA replication. Okay, so finally they concluded the mode of replication in which this DNA is getting replicated. So for this experiment, they took the E. coli bacterial cells as the model organisms, and they grow these E. coli cells in the nutrient broth medium, which is supplemented or which is enriched or incorporated with heavy isotope nitrogen, that is N15. They cultured this E. coli in N15 containing nutrient broth medium. So they cultivated it for many generations, so that the bacteria will take up the N15 molecule for its metabolic activity as well as for its replication. so that after many generations in their cell, it will have the N15 nitrogenase bases. All the nitrogenase bases will be converted to N15 nitrogenase bases. Okay, right. After cultivating these E. coli cells in N15 culture medium, they shifted this culture into another medium which is incorporated or enriched with light isotope nitrogen, that is N14. So N15, they have cultured the E. coli in N15 for many generations. After that, they shifted that culture in the media which is enriched with N14 isotope. Okay. So after shifting this, after inoculating this culture into the N14 nitrogen medium, they isolated the DNA from the cells in every generations. Each and every generations, they isolated the DNA and analyzed the DNA for the presence of N14 and N15 nitrogenase bases. Okay. They analyzed the DNA by density gradient centrifugation. So density gradient centrifugation, it's a centrifugation of the molecule with the presence of another molecule called cesium chloride. Okay. So based on the density of the molecules, it will be getting separated, in which the high density molecule will be separated or will be present in the bottom of the tube, whereas the lighter molecule will be settled on, will be separated, will be present on the top of the tubes. Okay. So based on this density, the molecules will be separated. So that for that purpose only, they have cultivated the bacteria in N15, then in N14 nitrogen containing medium. Okay, right. So after inoculating the medium bacteria in N14 N14 nitrogen containing medium, the zero generation of the cells were harvested. Few ml of the cells were harvested from the culture, and they isolated the DNA, and they go for density gradient centrifugation. Okay. So in density gradient centrifugation, in the zero generation, you can see all the DNA which is present in the bacteria is N15. Okay. So all the DNA which is collected in the zero generation of the bacteria is in the N15, which contains N15 nitrogen molecules, and the DNAs are separated in N15 band region in the density gradient centrifugation. Next, they isolated the DNA from the first generation after one generation. So since they know the generation time of the E. coli, they calculated the time when the culture is inoculated, and for the generation time of the E. coli is approximately 20 minutes. So for every 20 minutes, they harvested the cells from the inoculated tube and isolated the DNA, and they go for density gradient centrifugation and analyze the nitrogen, whether it is a heavy nitrogen or light isotope nitrogen by density gradient centrifugation. So in the zero generation, the DNA which is present in the bacteria which contains the N15 isotope nitrogen molecules. So in generation 1 also, they harvested some cells and isolated the DNA, and they went for density gradient centrifugation, in which they found a different result. That is, the band which they received does not correlates with the N15 as well as the N14. It forms an intermediate band. It forms intermediate band in between N15 and N14. It means the DNA molecule contains both the N15 as well as N14. Both N15 as well as N15 isotope nitrogen. In this generation 1 itself, they confirmed that the DNA is not replicating through conservative mode. If the DNA is getting replicated through conservative mode, it should contain both the N15 as well as N14 isotope nitrogens. But here, in the generation 1, it is having only one band. Only one band, that is in N14 and N15. Okay. So in the first generation itself, the conservative mode of DNA replication is excluded from the proposed model. And they analyze the second generation, generation 2. From this generation 2, after the 40 minutes, in 40 minutes time, after 40 minutes, they harvested some of the cells and isolated the DNA and went for density gradient solidification, in which they found two different bands. One is in the intermediate, that is N14 and N15, and another one is in the N14 band. That means some of the cells have completely N14 nitrogen bases, whereas some of the DNA in the cells which has both the N14 and N15 isotopes. Okay, thus they confirm this both the semi-conservative as well as dispersive method, maybe dispersive. In this second generation itself, they concluded that the dispersive method is not a functional one method of the DNA replication. They concluded the DNA is replicating through semi-conservative mode. Okay, so if it is replicating through dispersive method, means in second generation also it should have, all the cells should have N14 and N15 combinations. Okay, all the cells must have, all the DNAs in the cells must have N14 and N15 combinations of DNA. But here, some of the cells are having only N14 isotopes. Okay, from this they concluded this dispersive method is also not in action. Only the semi-conservative mode of replication is taking place. They concluded, and also they checked for third generation and fourth generation in order to conclude the semi-conservative mode, how it is. Okay, so in third generation, after 60 minutes, they harvested few cells from the culture media and they extracted the DNA and analyzed. In their analysis, you can see the band patterns. So the N14 band is somewhat thicker, whereas N15, N14 intermediate band is going lighter comparatively. Okay, so the proportion, the number of cells, the number of DNA molecules, hybrid DNA molecules are getting lesser, whereas the number of N14 molecules are getting greater in number. Okay, you can see, observe in fourth generation also after the 18 minutes, you can see the number of N14 DNA molecules are more in number, whereas N15, N14 intermediate hybrid DNA molecules are even getting lesser. Okay, so by this way, Meselson and Stahl, both the scientists have concluded that DNA molecules are getting replicated through semi-conservative mode of replication.